This is a production of Cornell University.
We'll dive in. So I wanted to acknowledge the other folks listed here as co-authors on this are graduate students that have completed their work at Cornell, working under this idea of further understanding far-red radiation. And then our funding sources are listed here as well. So why are we interested in far-red radiation? Maybe I'll back up for a minute. Why am I interested in lighting? It's because I work in greenhouses and controlled environments. And if we want to produce crops in the wintertime, we can heat a greenhouse, but that still doesn't make up for our lack of sunlight. And so we have, on average, maybe 5 times less sunlight, daily light integral in our winter days than our summer days. And so that implies our crop yields might be 5 times less, or at least significantly less in the wintertime. So then we need to add supplemental light. The LEDs have given us lots of opportunities to more precisely control the light that we deliver to plants, as well as giving us really great energy efficiency gains. Some of the best LEDs on the market now are about twice as efficient as the best high pressure sodium lights that we had. And so a grower could give the same amount of light and pay half the electric bill. In general, energy for heating and lighting a greenhouse is the second largest production input. And so that's one of the big barriers to profitability in this industry. This is a spectroradiogram of sunlight, and it shows us visible light from 400 to 700 nanometers, kind of from violet to red. And this is what we refer to as visible light. Interestingly, humans are really good at seeing green light. Maybe it's almost like we co-evolved with plants or something. So we're not good at seeing blue and red particularly well. So some of the newer like fire trucks are like this blaze yellow color or something like that. So our typical thought is that plants see all colors of light from red to blue and use that fairly well in photosynthesis. And that comes from the work of Keith McCree and others, where they would give single wave bands of light and measure photosynthetic responses on crops. And these were examples for crops grown indoors on the top and outdoors on the bottom. I think something like 27 different crops and relative quantum yield. And what we see is that indeed plants are pretty good at using all wavelengths from 400 to 700, but with a peak in red, a smaller peak in blue, and a bit of a depression in green, because plants reflect more green light than other colors or chlorophyll does, which is why they look green to us. But traditionally, we haven't thought of far red as being photosynthetically active. Far red will consider wavelengths of light from 700 to 750 nanometer. Many of you probably already know a lot about phytochrome. And so one of the pigments that plants use to perceive light spectrum is phytochrome. And one of the forms of phytochrome has a peak at 730 nanometer. So we think of far red as being 700 to 750 nanometer. Plants perceive the red to far red ratio. And so if they're growing in the shade, that implies that there's leaves above them that are capturing differentially more red and blue light and letting through more far red and green light down through that canopy. So this is a way for plants to perceive whether they're growing in shade or not, if they're high in far red. So far red in nature kind of triggers 2 responses, and really plants kind of fit along a continuum of these responses. It's not like they're either or, but kind of shade avoidance responses are where plants increase their stem elongation to try to outgrow the shade that they're in, as well as they might have earlier flowering initiation, and that might help them to reproduce more quickly if they're going to be under a canopy that closes up pretty quickly. They can also be shade tolerators, so they might grow leaves that are wider and thinner to try to cover more surface area of ground so that they capture the photons of light that do get through that canopy. So I would say that aspect of far red is quite well understood. However, photosynthetic responses of far red, if we look at the McCree curve, we would say, well, there's not much to this story. Why are we interested in far red in terms of photosynthetic activity? There is a tiny little bit here after 700. So one of the kind of critiques, and not real critiques, because McCree points this out himself in his work, is that they only looked at single wave bands at a time. What they didn't do is combine multiple different wave bands together. And so if we back up in our plant physiology knowledge, Emerson in the 1950s discovered what we call the Emerson enhancement effect, which is if you have wavelengths of light shorter than 680 nanometer, and then add some far red light to that it reduces some bottlenecks in photosystem 1, and you get kind of synergistic responses in photosynthesis. So our group, as well as largely other groups, have worked on redefining the definition of what's photosynthetically active, and they're proposing that within bounds, we think of all light between 400 and 750 nanometer as being photosynthetically active. Dan tells me I have 5 minutes left, which seems impossible, but I must be true. We've been studying this quite a bit with lettuce, and one of the things that we did is, so if we consider far red photosynthetically active, we wanted to do a study where our control white LEDs, which have only 2% far red, and then we added 20% far red in another treatment, which is closer to the sun. Sunlight gives us 14% far red, roughly out of its total light. So we wanted to apply the same number of total photons of light to lettuce plants. And then we sampled them and looked for what happened at both the seedling stage and the finishing stage. So at the seedling stage, the plants with high far red are on the left, and control plants under simple white LEDs are on the right. And indeed, we see more leaf expansion, more leaf area. And then if we transplant those and grow them out to a final harvest stage, we see similar continued growth benefits for lettuce plants. So we saw something like a 70% increase in leaf surface area, and then similarly fresh weight and dry weight were also 70% larger. So we applied the same number of photons of light, but we got 70% more biomass when we added far red. And then we were curious as to, so if we thought about the leaves themselves, is it due to an increased expansion in cell size, or is it due to an increased number of cells within each tissue? So we carefully, Nate, the grad student working on this, carefully looked at several different tissue types in leaves, and these were results for epidermal cells. So he looked at what we call leaf 5 at day 23, and indeed found that epidermal cells had a much larger surface area under far red on the left side versus 2% far red on the right. However, he also did calculations based on, we knew the surface area of each leaf, so we could calculate the number of abaxal epidermal cells in each leaf. And we found up until about day 25 in development, we did see an increased number of cells themselves. So it wasn't just cell expansion that contributed to this, it was also an increased number of cells in that line. We've also looked at far red from the standpoint of flowering effects, and petunias has been a nice model system for this. This was kind of some preliminary research. This is an obligate long day plant, and it was grown under 16 hours of light. And under either low daily light integral or high daily light integral, if we had very low far red, we saw huge delays in flowering. It took like 200 days for these plants to flower. And then under 20% far red, they flowered under a much more normal timeline. However, they were quite a bit taller, so they're showing those shade avoidance responses. Now, 2 minutes left. Okay, so Darren screened 5 different cultivars of petunias for flowering responses and other morphological responses. Under far red, varying from 3% on the right side, that was kind of our control white LEDs, all the way up to 22% on the left side. And we do see larger plants, larger leaf surface area, but maybe more dramatically, we saw 1 to 3 weeks earlier flowering in petunias. In some cases, also excessive stem elongation. And maybe I'll skip the hemp research, but I'll also point out we've been studying far red in hemp, high cannabinoid hemp, so high in CBD or CBG. And essentially we find taller plants, but no benefits in terms of biomass or cannabinoid concentration. So to summarize our work, we do find, as well as others, that what we call extended PAR may be a better predictor of crop growth than photosynthetically active radiation. Lettuce is representative of shade tolerators. Hemp and petunias are representative of shade avoiders, although really plants can kind of be placed on a continuum between the two. And we see that there's greater need for research on the far red fraction that's beneficial for photosynthesis. So that might max out at 20 or 30 or 40% of total light, affects on morphology, species, and cultivar. We need more research on how far red impacts or affects other environmental parameters. So under higher temperatures, we see less of a far red effect because temperature can also affect phytochrome photoequilibrium. And we're starting to find effects on plant water use efficiency, where far red decreases water use efficiency, interestingly decreases uptake of iron and manganese, so that could affect nutritional quality a bit. And so we also need to understand post-harvest quality as well. I'll stick around to answer any questions later so we can move on to our next presenter. Thank you.
Yeah, thanks a lot for the introduction. Just one small correction. I actually arrived this year in January, so I'm brand new. So it's great to be here. I was really excited for this opportunity to kind of introduce my research program, but also because tomorrow's Halloween. And spooky season, I think, is the perfect time to celebrate the herbarium, of which I am the new director of. Here on this intro slide, I'm showing just 4 example specimens that we have in our botanical crypt, which basically stores these dead, pressed, and preserved plants for centuries. These 4 plants happen to all be collected on this day, October 20th, but over the last century. And so here are examples of some other collections that we have in our L.H. Bailey Hortorium Herbarium at Cornell. The specimens along the top row are all plants that were collected this year. And most of these specimens were actually collected by our biodiversity interns, which is an opportunity for undergraduates during the summer to gain experience in plant collections, but also herbarium practices and research applications using the collections. So keep an eye out for that opportunity for interested students. The specimens on the bottom row are collections that were made 100 years ago in 1925. And so you can see, even across a century of time, and even though that these plants are pressed flat, they still capture a lot of attributes of the physical characteristics of the plants, their architecture, and even preserve, in a lot of cases, the colors really well. And so I think, these dead plants, in some cases, are even more beautiful in their pressed state than they are when they're alive. So herbaria are more than just a collection of dead plants. These physical specimens, do make up the core of the herbarium. And each physical specimen represents an individual plant that was collected either in the field or from a greenhouse at some point in time, pressed, and then brought into the herbarium for processing and long-term preservation. But along with each physical specimen, we have a lot of associated information about who collected that plant, when it was collected, where it was collected, and even sometimes a lot about the environment around that plant, including abiotic and biotic interactions that were occurring at that moment in time and space. And so both our physical or all aspects of our physical specimens are in the process of currently being digitized. And so we're taking images of all of our herbarium sheets, and we're also transcribing all of this associated collection information from specimen labels and field notebooks, and making all of this information available in publicly accessible databases. So you may have heard of things like GBIF, right? Importantly, both the physical and digital aspects of biological collections are incredibly useful for different questions in research applications. And together, they constitute this archive of phenotypes to genotypes that are preserved through time and allow us to address a lot of different questions that really form the foundation of plant biology. And another important aspect of herbarium specimens is that because they were collected by humans, they really span the range of intensified impacts that we have had on our planet. And so ancient DNA can be retrieved from these specimens and provide really unique glimpses into interactions across the plant community between the plants and the environment and other species. And we can also see genetic changes over time. And so in my lab, we're really interested in studying ecological and evolutionary processes that are shaping biodiversity across spatial and temporal scales. In particular, we have been focusing a lot on the diversity of plant communities and high elevation alpine ecosystems. And ultimately, we're really interested to try to understand the fate of these plant communities as both the environmental as well as biotic interactions continue to change. And so because we're interested in change, we rely a lot on both the physical as well as the digital components of herbarium specimens to address different questions about biodiversity from multiple perspectives. So these are just some examples of research in my lab from the last few years. So we extract DNA from vouchered specimens, especially from plants that are sometimes really challenging to find living tissues for. So these include rare specimens, plants that have actually already gone extinct, or plants that live in challenging to access areas like alpine regions, right? So we extract DNA and RNA molecules from these specimens to try to understand things like cold adaptation using comparative phylogenomic approaches. We look at genetic change over space to try to understand local adaptation in extreme environments. We look at images of herbarium specimens and categorize and measure different traits of the plants to try to understand how things like climate change are impacting shifts in plant flowering time. And more recently, we've been trying to extract other types of communities from the specimens themselves, including viral communities, and we're interested in exploring pathogen dynamics. Finally, we spend a lot of time in my lab trying to synthesize all of this biodiversity data that's available out there. And we develop tools that allow other researchers to be able to look at overlap in spatial and temporal depth to help them to identify plant collections or areas where plants have been collected that might be able to best inform their research interests. And so I'm happy to talk about all of that more, but today I'm going to focus a little bit more on how we can use herbarium specimens to unlock new discoveries in the future and some things that I'm really excited about that are currently being developed. So while the scope of research using collections historically has been really broad, applications, arguably with kind of more direct impact on humans, so applications surrounding research in agriculture, public health, disease ecology, have really been lagging in the realm of utilizing biological collections. And so I see a lot of opportunity here to try to diversify uses of herbarium collections through new research innovations as well as novel research frontiers to try to improve human well-being. And so I think a lot of the future discoveries from these centuries-old specimens will really be driven by a lot of exciting new technologies that are being developed right now, first of all. And so a few examples I'm really excited about are ongoing developments in next-generation sequencing. And so these developments are allowing us to increasingly more cost effectively and efficiently extract both DNA as well as RNA from fragmented genetic repositories in these historical collections. And there's been some really exciting research that is expanding this across many different samples to explore things like shifts in weed adaptation and range expansion across agricultural communities. Hyperspectral scanning is a technology that's often used in field and laboratory applications, but there's a lot of ongoing research trying to develop this as a tool to rapidly and effectively measure different types of traits from pressed plants. And then last but not least, of course, artificial intelligence. Biological collections are a little treasure trove for AI. And so both the image of our specimens as well as the digitized associated collection information provides a lot of resources for computer vision and language learning models. And so there's been a lot of recent interest in AI applications for biological specimens. We're already using AI a lot to make our lives easier in terms of digitization and things like this. But the path forward is trying to figure out how we can use AI for sort of high throughput phenotyping to work through the hundreds of millions of images of specimens we have held in databases representing collections around the world. And so these new frontiers are addressing pressing modern issues, everything from conservation to human health. And so these are just 3 quick examples, but there are really so many more. And so just some quick ideas is that there's been some really cool new research actually isolating nodule-associated bacteria from centuries-old specimens and creating cultures of these, sort of like a time machine, bringing them back to life. And there's a lot of ideas of how to kind of use this to explore spatiotemporal plant microbe interactions. We have AI approaches that are training to try to better detect herbivores and pathogens from plant images, and then also using images to try to understand the timing of pollen release and inform allergen predictions and improve public health. Right, so we know the plants that provide important services for us, but we still have a lot to learn about them. And so I really think the future of research using our collections in the herbarium that are increasingly being digitized is really thinking about how we can apply research from these collections more directly and immediately to the plants that we rely on for things like medicine, food, materials, and our general well-being. And so maybe the solutions for sustainability are actually held in our herbaria. And right here at Cornell, we have an amazing herbarium with lots of untapped potential. So our herbarium here holds almost 1 million specimens collected from around the world. Most of these, or a good portion of these, represent plants native to New York State, but actually our largest collection are horticultural and agricultural plants. So we have about 250,000 species that really reflect the history of horticulture in the United States. And really nothing has been done with them. So besides these plants, we have a lot of other associated collections linking to them, including seed reference libraries, horticultural catalogs, other images, and tissues preserved specifically for molecular research applications. So we are doing a lot of exciting things in the herbarium, and I'm excited, in my new role as a herbarium director, to facilitate all of these future uses. So please reach out to us if you have any questions. An exciting piece of news is that we're hoping to announce the release of our own digitized database representing what we have digitized so far from our collection, so it's much more easily searchable. And that should hopefully come out by the end of this year, so stay tuned. And with that, I'll leave you with some more fall foliage. And also, I have treats, which are herbarium stickers. So I'll pass these around while the next speaker gets set up.
I want to start with the basic premise that I hope I don't have to convince anyone on, but I'm happy to convince you outside of the seminar if you need, that gender inequalities are pervasive in agri-food systems globally. We have enough evidence, decades of evidence who've documented this. So we know that there are a lot of things wrong with the agri-food systems in terms of gender inequality. So this is our premise of why we do the work we do. Coming a little bit deeper into this, we look at across agri-food systems and we define them broadly as kind of value chains where you produce, process, and market any crop to the food environment and consumer behavior. A lot of the work that we're seeing shows that women's agency or their ability to kind of bargain and have their own life aspirations and fulfill them together with gendered social norms, policies, and access to control over resources, all together shape how they interact with the food system. Okay, again, these are things that we know. So jumping through a very big logical leap into why we do this in plant breeding, I'm going to kind of set out this basic definition, which I love to use, that is plant breeding is a genetic improvement of plant for human benefit. And I love this quote because it says human benefit, and the question I like to ask is which humans and what does benefit mean, right? How are we defining those humans, who they are, how we're engaging them, and what benefit means to them. And this matters because if we look outside of agriculture and agri-food systems, if we look at technology, science, and design processes, there's a lot been written about what's called the quote unquote default male being what is at the center of innovation, right? There's a blindness, there's a data bias in terms of what we know. There's a lot of different websites, a lot of different examples I can give, but if you want to dig in your own time, there's whole websites called Gendered Innovations that look across different sectors to show that lack of data on women and people of color really have shaped outcomes of technology, okay? And this holds across many different sectors. AI, you've probably heard a lot of the facial recognition software and seatbelts and things like that. And this is because as scientists, as we do our work, we're not doing it from a position of complete objectivity, no matter what we tell ourselves, right? We're shaped by our social interactions, we're shaped by our upbringing, we're shaped by who we are as humans before we become scientists. And it matters, and there's a lot more work on this coming out, that acknowledging these kind of systematic inequalities of who's involved in research, who's doing research, how and why, and the outcome of research is just good science. So recognizing what we're doing and how we're doing it, doing that better is good science, okay? So I'm going to walk you through a quick theoretical framing because we need to do this at this stage. So I've talked about contextual factors. Take this as social context, right? As humans are producing different foods and they're engaging with agriculture, who they are, what markets, their demographics, what markets, what environment, what norms, together with what resources they have to produce food are all shaped by gender inequalities. When we take that a step further to plant breeding, we know that these resource imbalances and contexts shape what individuals particularly like or need from the crops that they're growing. Okay, call this trait preferences. And in turn, how we do breeding or the institutional way that we do breeding is also shaped by society and social inequalities. So together we think about how we treat gender inequalities in plant breeding. We look at the traits, but we also looked at the act of plant breeding to say the new breeding products, new varieties, so to speak, have to take into account all of this. Okay? It's a large ask. But at the end of the day, we want breeding products and new varieties that take into account inequalities, and we want them distributed to be able to get to farmers' fields, to have actual outcomes. Okay? This is a global framing. So what do we do in our lab? Basically, we bring this piece, which is, if you look at gender inequality, all the multilaterals talk about change from the individual, household, community, organization, macro-environmental. That means that is not the responsibility of individual women to make societal change, but we need to think across these different scales, okay? And at the same time, we know how we need to do plant breeding differently. So our work is to bring those into conversation. And I'll explain what that looks like in a minute. So the work that we do is really to build crop improvement projects, programs, and institutions that center on gender inequality. Okay, it's not an afterthought that happens after, but we're really intentional in how we build them. So we do all sorts of work along this spectrum that I won't dig into in an interest of time. So you're probably thinking, what does that actually look like? What does research look like in this space? So I'm going to give you some examples of the work that we do. I've done a lot of work over the years on cassava and cassava seed systems, but really the gist of it here across many different grants that we've been involved in over 10 years is trait preferences. So we've done a lot of work, not to just say, what do farmers want from cassava? That's a big question, and the answer is simplistic. We've taken that a step further to say, what do women in the southwest of Nigeria living in poor and food insecure households want from cassava? That's a very different answer to a generalized question. So what we've tried to do is really generate nuanced evidence to bring social context into the picture, to understand how improving different traits will have better outcomes on poor women in food insecure households, for example. So prioritizing what a program does with this framing. So there's a ton of work. I'm welcome to answer questions on what we've done in that trait preference space. But I'm going to draw attention to some newer stuff that's coming out that I'm really excited about. And this is in relation to here, right? So I told you that social context and individual context matter with each other. We've done further work methodologically on this because it's not easy to capture this. You can't just walk into a household and ask people what they want, right? Methodologically, there's a lot that goes on behind the scene. So I collaborate a lot with economists, and this was a work with a Dyson student and a postdoc in our group that we looked to how to do a choice experiment. This is giving people choices from different things and how to understand them. And basically what we did here was we started from individuals living in households. This is work with Israbame in Senegal, and it's working on cowpea. So we had individual characteristics of people living in a household, right? These are spousal couples in this case. Then we had how they were interacting. So we knew their age, education, so on and so forth. But we also knew how they were making decisions. What that means is who's making decisions on cowpea production, who's putting most time into the field, who knows most about the crop. And we generate through various mechanisms what we call concordance. All that means is the husband and the wife both agree that the woman has the decision-making role. That's a proxy for empowerment. That means that that woman is more empowered, okay? So we generate these kind of indices. And then all we want to do at this point is make very complex data tell us how choices are made. So what we're trying to do here is understand how individuals within households make trait preference decisions. This is very messy. Don't worry about the bars, but I'm going to give you the bottom line here. Basically looking at an individual within their context, right? So a woman in that household that may be poor, is market-oriented, and is relatively disempowered. What does that mean for trait preferences? So we have 5 traits we're looking at here, again, cowpea and Senegal. So methodologically, we were able to kind of innovate on how to understand these households and individuals. But what we saw was really interesting as women became more empowered, and we're calling that concordance. So there's agreement of their empowerment. They make more specialized choices. And this was interesting because for the breeding program, this could be important as they're deciding which trait to really prioritize. So we're giving them that information. A second example is work that we're doing with Common Bean in Costa Rica. And the starting point for this study was interesting. It was through the Feed the Future Innovation Lab for Crop Improvement. And the breeding program simply asked us, I want an adoption study. I want to know who's growing what, where. Very simple. Okay, and there's long-standing econometric methods to do this with. But what we wanted to ask is, does it matter who you ask what they're growing? Okay, not just saying people are growing this. So the latest, greatest way of measuring adoption currently is using fingerprinting. Okay, so as researchers will go out, take a leaf tissue and say, this is the variety that's being grown in this field. But what we wanted to test was, instead of going straight to the field to take leaf tissue, which is common practice, what happens if we make different decisions with who we go to the field with? So to oversimplify it, we did kind of a knowledge index or information index. If 2 people, 2 adult decision makers, had the same score, we went with a person on a farmer-grower list. If one person was higher, we went with them. Very simplistically, there's a lot behind this. What we saw was, if you go, and this is a moot point, but here it is empirically, if you go with the person with more knowledge, you get better agreement between the genomic data and the self-reported data. This has never been shown. It's an assumption, but we're able to show it. More interestingly, the more variability we had in the reference library, it was harder to have a match. And that made it interesting because there's narratives around farmers don't know what they're growing. And we're pushing back here saying, how about, is it the way we're doing these studies that may be explaining some of that error? So we're kind of questioning the error in the system. Okay, I'm not going to dwell on here. We do a lot of work in seed systems in Tanzania. So this is a seed systems model that's kind of decentralized, using community members to multiply seed and sell to their neighbors. We're doing that with a really strong gender intentional lens to how to empower women within those seed systems. This has a website if you want to look at in particular. And lastly, kind of at the organizational level, we've been doing a lot of work over the last 2 years looking at how women are perceived as leaders and managers, and how care responsibilities of women and men and working couples play out across plant breeding fields. Really interesting findings on how we do the research, right? Who we are within our research fields. And lastly, we kind of look at the larger level, looking at donor bias and seeing who's funding what, and the same idea of gender blindness that we see in other fields is playing across our fields as well. So we're demonstrating that through data. So closing thoughts, gender inequality in varietal design, development, delivery is really complex. So we need to bring complex thinking and interdisciplinarity. More and more our lab works on methods because it's really hard to bring different disciplines and do methods together. So we spend a lot of time on that. I think it's really important to do contextual research, but also scalable approaches. And this is probably something we talk about most in our lab is how are you in a project and at the same time critiquing it? So having that inside-outside focus. So at the end of the day, my pitch is thinking about power and politics of science is as important as the science that we do. So with that, thank you to the lab members and our collaborators. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.